I feel so violated.
Such a graceful dance.
I'm having a little trouble with the autofocus here on the, all these seem very blurry.
On the cinema cam?
Yeah.
Yeah.
I'm just like reviewing the images after and they all seem a little off.
I can come try to help you with that when we're...
Did you maybe toggle the autofocus off and try to manual adjust and then toggle it back on?
A viewer is asking, the purple color, is that a defense mechanism?
Do you mind if I zoom
Oh,
in?
no. They just, they come in purples and pinks, and it doesn't really help them or hinder them in any way. Most things don't really want to eat them either. They just look fabulous. You can see right into its mouth.
Yeah, and if you turn the knob while it's autofocusing, it won't change. You have to be toggled off.
So I'm off now. No, it's adjusting.
Nice.
And then, but that should autofocus, right? Is that right? Because yeah, but when I review all these images, they all seem a little off.
It's tricky.
Yeah.
It's quite sensitive to movement, these cameras.
Yeah, we might have to have a screen with that because it's very hard to see up there.
Yeah, yeah. Especially when you're zoomed in, when you're fully zoomed out, you're looking at a lot. It can focus a lot easier, even when moving a bit.
Yeah.
Some of the imagery on our first dive from the WCA is really excellent. Yeah.
A viewer wants to know, how does a sea cucumber taste? Oh, this one? Not very good. It's mostly water. Tastes like seawater.
What's your favorite way to prepare a sea cucumber?
I don't eat sea cucumbers.
Okay.
Alright.
I think
What
they
the cucumber
taste
be?
pretty good. I mean, well, not deep sea cucumbers because haven't tried those. But like the ones from the seagrass beds, like depends on what part you're eating or what species you're eating. Some people eat intestines. I personally don't think it's great. Just trying to, but other ones they eat the flesh and those are pretty good.
that about it on time on bottom, Daniel?
No, I had it wrong. So I think, yeah, it's at 38 or 35 past the hour.
Okay.
Yeah.
Bridge now.
So yeah, 14 minutes.
Every last minute.
Yeah, exactly. It's so rare to have these opportunities. We had a couple of ways that we extended this dive. So we were originally planning to do the personal transfer with our boat, and that would have taken several hours to run back in and then come back out. Instead, we are working with a company basically to just, with a larger speedboat, just meet us out at the pilot station, and that saved us about 4 to 6 hours. So that's why we were able to extend this dive by another 4 to 6 hours, and especially when you have these deep dives where it's 2+ hours of descent and ascent. Because it's unlikely that there'll be a dive in here at these depths anytime soon in this very area. But yeah, I'm very, very happy that we covered that much horizontal distance.
Oh yeah, there's a bunch of things on that
Yeah.
stock, yeah. I think there's cereanthids.
Push in for a zoom.
And there's some polychaetes and barnacles as well. There's a lot of barnacles.
Yeah, that's great.
Nice.
You can see more of those circles in the bubble cam view.
Mm-hmm.
It's kind of a good angle.
Oh, there's an anemone on that stalk.
Yeah.
It's a hormathiid. There's another one of those things. The cucumber, maybe, on the ground.
Go for a zoom.
Oh, and there's a crinoid too, and some gooseneck barnacles.
Yeah, and isn't that like what we collected earlier?
Yeah,
Right underneath it, yeah.
there's another one of those things that we collected earlier. Oh, there's a really cute little ice pod. See it with its little
Yeah.
legs hanging out. It's in the family Arcturidae, that isopod. They usually do that posturing. I've seen them on corals and sponges and things.
The anemone is likely in the family of the Amphianidae.
Oh,
That's what Chris says.
okay. Well, the amphibanthids are usually more smooth. So this one was looking more like a hormathiid.
Yeah, I
I'm
agree
not
with
sure.
Megan. They have a lot of bumps there.
Yeah, but both of those tend to have that sort of Venus flytrap kind of look to it. Bridge now. Can we make 40-meter move to 90?
Another dermathiid cucumber, no need to zoom in.
Gosh, I don't think we've seen a fish.
No.
No.
Yeah, not on this watch. Another dermathiid sea cucumber.
Oh, there's a fish.
Oh,
Yes.
Sarah
Had to
manifested
speak
it.
speak it into existence.
Yeah.
I know.
Oh, no.
Oh,
It's like, I think not.
What kind of fish are you? Like, we can't tell.
Oh no.
I'm thinking halosaur.
You get the end of your tether too.
Yeah.
Yeah.
It's moving like a halosaur.
Oh, alright. Picture here.
Oh, yeah, it was a halosaur.
nice.
Alright, we got one.
we got a fish.
What else do you want to see, Sarah?
So I've yet to see an anglerfish. I'll like go off watch and then the next one will be like, oh my god, we saw like 2 amazing ones. I'm like, ah.
A viewer wants to know, what are the chances of seeing any octopus in this area?
An octopus?
Mm-hmm.
I mean, like, it's likely. I mean, they're probably around here. It's just, Luck of the draw.
Look at those sediment patterns in the WCA view.
Oh, yeah.
I
That's
wonder what
weird.
that is off to the port side.
It's like it slumped a little bit.
Yeah, I guess we're going up a, oh yeah, yep. We're going up a steeper slope here.
You know what we probably will see? Some squids on recovery.
Yeah, speaking of which, I think we'll probably call it here.
All right.
So we'll have 4 Niskin bottles on the way up at 1,000, 500, 250, and 100. But yeah, it should be about an 82-minute ascent. Yeah, I just want to take a moment, thank all of our viewers and all of our scientists ashore for joining us, and everybody here in the control room for all their hard work. For those who are interested in the water column ascent, there'll be about an 82-minute ascent to the surface, going across 2,400 meters plus of blue water. And then the next dive should be deploying at about 26 hours from now. It's on an unmapped seamount on the eastern corner of the exclusive economic zone of Palau, within the Palau National Marine Sanctuary, on a seamount that has not yet been mapped. We'll spend about 8 hours mapping the entire seamount, and yeah, for those who are scientists ashore, we'll try to set that dive plan out to you as early as possible, but it'll probably will be about an hour before we dive, just because we'll have to finish mapping it, and it'll be kind of a last minute, so we gather as much possible information before we compile the plan. And yeah, thanks so much for joining us, and hope to see you again tomorrow at this little after this time.
All right. Ready to start coming up. Thank you. I might need to pitch a plate to make it on time. All right. Yeah, we're only making like 20 meters a minute, so. Yep. All right. Almost 24 meters a minute. Yep. Keeps me focused.
It keeps you focused. I feel like I would lose focus if I was cold.
I make it a point in Rhode Island to wear shorts as long as possible. Long into December. Shorts and Crocs.
Yeah, if my feet got cold, then I would just be done. Crocs are just like shoes that have just holes in them. How do your feet stay warm? Mm-hmm.
Yeah.
Mm. You could 3D print them.
You can.
We have TPU. But they wouldn't be as cool as Jake's Crocs.
I have little flashlights for them at home.
Really?
Yeah, you stick them in and they're little like headlights. I forgot to, I was gonna bring them out, I forgot. But
Feel like that could come in handy. Like if you were like maybe cleaning underneath somewhere, you're trying to see. Wake up in the middle of the night. Are they motion activated?
no, they have little buttons. So.
Did you ever have light up shoes as a kid?
Oh yeah.
Yeah.
Oh yeah.
Freaking love light up shoes.
Okay. You can speed up a little bit on the winch. 27, 20. What's our first Niskin depth?
1,000.
1,000. All right.
That one spins good.
Mm-hmm. This one's got good bearings.
Mm-hmm.
It has.
And when we're on bottom, I feel like the time passes too quickly. And then you get in the blue water, like every minute is like an eternity. You look back, you're like, oh, well, we've only ascended 10 meters since I last checked.
Blue water.
Blue water. We had a whole blue water watch earlier. That was intense.
That's better.
Yeah. Mm-hmm.
Yeah,
Herky jerky.
Nice. Yeah, let me play around with it. Yeah, you gotta... Yeah. Pan and tilts. Uh-huh.
Yeah. Oh,
Could we get a Niskin sample? Oh, thank you.
That was in number 3, right? Cool.
Hi, Team 12 to 4. Just gonna come interlope with y'all for a bit.
Hi.
A viewer wants to know on channel 1, what is the V-shaped green dot? Somebody want to explain for me?
That's the lasers.
Tell me more.
They're mounted on the HD camera and they're 10 centimeters apart. We use them for scaling different organisms while we're getting good zooms on them.
Jake, where exactly are they, like on the camera?
Mounted on top, kind of like a pair of eyes, looking straight out.
Do they come on and off each off season? Do you have to recalibrate them?
We have a mount for them,
Okay.
and that mount is 10 centimeters. It keeps them 10 centimeters apart. But we do, yeah, we remeasure it during installation. Sometimes the lasers themselves need replacing, so they're actually just basically laser pointers that you'd use every day for
Your regular
yeah,
laser
regular old
needs.
laser pointer, like, you know, as a cat toy and stuff.
For all your standard laser pointer.
Yeah, they're just in a housing that protects them from the pressures of the ocean.
And why do we choose green?
Green because red light travels the least in water. It absorbs, it attenuates. Green travels a bit farther, and it's also distinguishable from blue. Blue is the light that travels the furthest in the water. That's why the water kind of appears blue mostly. And when things get farther away from the camera, they appear blue.
I got this question the other day, and I didn't actually really know the right answer. Do you have a sense of how far out they travel?
That I do not know. But I don't know. Normally see the
Quite a
lasers
few
on things
meters.
quite a few meters away. Yeah.
Yeah. We could be, 7 meters off bottom and still kind of see the lasers.
Yeah.
Now, you won't be able to measure anything from that distance, but you can see them there. The green ones you can definitely track. With the red lasers, it's a lot harder to track from further away. And a lot of the things that we look at tend to be like pink or purple, which makes using red lasers challenging. Nothing that we see is like lime green, so it makes it easier to see.
Nothing yet. Megan, is there a good, or anyone, is there a good answer about, we get this question a lot from kids, curious, like, why is everything pink in the deep sea? There's a lot of pink and orange and red.
So a lot of things might have red coloration as a way to blend in. Like Jake was saying, red light attenuates first in the deep sea. So if you are red in color, you're essentially black and very challenging to see you. And you see that most often in the water column. Things like your corals and sea cucumbers, they might have various colors, and those colors are due to different things, chemicals in their bodies, and they haven't really been, evolutionarily removed because it doesn't harm them to be that color in the deep sea. So while they don't need to actually blend into the dark, they can have these colors for other reasons, but not necessarily as a way to use color in their environment. It could just be a chemical that is used in their body in some other way, but gives them color. Some other animals could have color because they were that color at shallower depths, and when the animals moved down to deeper depths, evolutionarily maintained that color. There are a lot of theories about color in the deep sea. We can't know all of them for sure. Every animal is different in their reasons for having that coloration can differ.
Like you said, there's not a lot of lime green. Are there any other colors that you feel like we don't see that often?
You don't really see like bright blues either. There are some animals, like Rexia can be blue, but...
We found those like blue purple plexaurid at Jarvis a few years ago, and
Yeah.
it blew my mind because I was like,
That one sticks
it blew my mind.
in my
Yeah.
memory too.
I was really hoping that we were going to have the opportunity to witness that again this year, back down at Jarvis.
Yeah, I was too. I was very disappointed. Maybe another year.
Yes,
Maybe another location.
absolutely.
Now, there was a crinoid that we picked up. It was shallow in American Samoa. That was a very interesting green color. It was like green and black.
Oh, wow.
You tend to see a lot more variation in color at shallower depths, but there is a lot of light there, so a lot of those animals use color in a useful way, whereas in the deep sea where it's very, very dark, coloration isn't as important. But you'll also notice that a lot of the animals down deep still have eyes, and that's because a lot of animals bioluminesce, and so they can use their eyes to view the bioluminescence. Reasons to produce bioluminescence vary. Some produce bioluminescence as signaling for mating. Some do it as a defense mechanism or a combination of those types of things.
Finding friends, finding food, fighting friends, fighting food. Yeah.
Some fish use it as counter shading to blend in to the environment so that predators from below can't look up and see them as easily. Breaks up their silhouette.
All right, science team, back row, got a question. What happens with our samples after the expedition is over?
Well, it depends on the expedition, but for this expedition, most of our samples will be heading off to...
Oh, yeah. Yeah, why don't we, go down and take a look at it. I think Mike's up too, so you can get a second look.
Yeah, so most of our samples will be heading off to our two repositories.
Yeah. Okay.
The Museum of Comparative Zoology.
I think that's always like that.
Yeah.
Harvard University, which is essentially a library of samples collected from numerous expeditions over the years, and they have facilities that can maintain these collections and databases and also share them with scientists around the world. So send them off on loan, just like checking out a book at a library. And we have a similar repository for our geological specimens at the University of Rhode Island Graduate School of Oceanography. It's called the Marine Geological Samples Lab. So we'll be sending our rocks, and if we collect any sediment cores, those will also be sent there. Those are our two primary repositories, or sample libraries, that we send all of our samples to across our expeditions. But when we have collaborators on board with us, sometimes samples will go home with them, and for this expedition, all of our eDNA filters and samples will go directly to Pickrick, if you want to share a little bit about that process, what you'll do with them when you get back to the lab.
Yeah, so we filter the water samples on the ship. We bring them back and we send them to our collaborators from Stanford Center for Ocean Solutions, and they extract the DNA, and that tells us which fish, because we mainly look at vertebrates, so like which fish lie within PNMS.
Niskin 4.
So Mai, tell me more about that process. Like what, is the lab analysis done on island or with partners?
The analysis is done with Colin from Stanford Center for Ocean Solutions.
All right, triggered.
Yeah.
500 meters.
And then they are
Copy,
sent
thank
to
you.
a third party for sequencing.
And then what's that timeline like? When do you usually get some results back?
It takes a good while. I think we just got results back a few months ago from samples that we had in 2021. So it takes a really long time.
Oh, wow.
Yeah, just because we have to send them there, and then because we have many replicates for each site, it takes a while to extract DNAs from those replicates, and then they still have to be sent for sequencing, which takes a while.
You can go down. Oh, Mike is. Okay.
Brian, can you speak to that? How kind of normal is it that, you know, I think about this being like the very start of a very long road of science that will come afterwards, this like 10 days, but can you talk
Unless
about
he
those
says stop,
timelines?
I'll
Like
keep going.
when do we start getting, when do we expect to start getting some results and kind of rolling out this knowledge?
Yeah, it can be a very long timeline, and it varies greatly. It can take anywhere from just a few months to a decade plus sometimes, and even longer. I mean, I've been doing an analysis that I've been digging back to records from the 50s and older.
cool.
So it really depends, and sometimes you end up with sample analysis from the 1800s. I've seen a couple people trying to figure out the range of certain taxa, digging through the museums and finding like original Androagases, collected coral skeletons from the 1800s and stuff for use. So, you know, science is a progress, and we all stand on the shoulder of giants, and it can be, you know, your work can have impacts hundreds of years later potentially, but generally a few years. So I've like in the process now of just wrapping up a project that we did the field work in early 2021. I'm going back and forth with a journal about edits and reviewers right now. So 2 to 3 years is a pretty common length from data collection all the way to publication. But, you know, that's not even the length of the project. The length of the project really starts from the funding and the writing the proposals and trying to figure out where you want to go. And so that's another often year to year and a half, even 2 years before the project, that you have the idea, you start writing the proposals, you get the money, you start working on the ship time, you start working on the permits, and then all the logistics that it takes to mobilize a ship somewhere remote in the world with a team of 50 people of all the different components of driving the ship, making sure you've got the food, making sure you've got the equipment and the supplies, making sure you've got the expertise. So it is a multi-year project. And you know, you've got mainline projects that you kind of start from the proposal stage and work all the way to conclusion. Then you've got all the side projects where you're at sea and you find something interesting that you aren't necessarily funded to work up or don't have the expectation to do that analysis, and you have to find the time and the expertise to do that kind of on the fly, and the idea hits you. So there's no straight answer, but it's usually definitely on the order of years from, you know, when you're out here at sea till when a published piece of academic science comes out in the literature.
Is there such thing as like a total species inventory for Palau National Marine Sanctuary? Yeah. Like, do we have a master overall list that we're gonna be like adding on to from this expedition?
We currently don't, but that's something that we totally should do. I think like when we get back, I'll talk to Igalao about it.
Yeah.
Because it's definitely something that like good to have for reference for like people doing research and just like curious students. Also really good for outreach stuff.
Yeah.
So we definitely should start one.
I feel like it would, it just like the title of it should be giant asterisk, like, here is the very beginning of a list of which 99.9% of the list is not here yet,
Yeah.
But
Yeah, that would be quite a task to... It would be quite a task to go identify all the samples across all the various expeditions over the years in an area. I know that expeditions like this where the data is really public and the whole process is really public, it's easy, but who knows about other sampling efforts across other time periods and expeditions. Some of that information might be a little bit more tricky to track down, but
yeah,
it'd be really, really amazing to get.
we definitely know, I mean, that like process of unearthing, revealing data that might be on somebody's hard drive somewhere in their desk, like, is a huge part.
Having worked in both of our sample repositories, MCZ and MGSL, in the past, I always really love to see some, I don't know if unusual is the right word, but unexpected requests come in from researchers around the world asking for some specific sample that hadn't been touched in a long time, and you just never know when something might be really useful for a new kind of archival study or project. And that's how these archives of samples can be useful for decades and decades to come. Often we collect so much data that it can be already just a lot to work with the new samples that we have, but there's just so much there in the archives that there's just quite a lot of work to be done. So,
and it's oddly hard to get funding for archival research and analysis of existing data sets. There seems to be a bias in the funding agencies, and at least in the United States, that are kind of biased towards fieldwork. And so every PI lab researcher I know has data unanalyzed or samples that they know are in the archives or in their own lab that were the side projects and were extra things that they'd like to work
let's be
up,
nice to the bridge.
that they don't have the funding or the time to do, and yet they're still planning more field work because that's kind of the easier path for getting large grants. I'd really love it if we would see more data rescue and existing data set money out for researchers to work with.
Yeah.
yeah, that's a really interesting point. Some of the...
May is 250 meters our next Niskin?
Yes.
Okay. Cool.
250 and 100. Yeah, some of the archival studies I'm aware of from researchers and former advisors of mine found clever ways to link archival samples and work to future or planned fieldwork as well, to kind of extend maybe some collections in order to better understand a more regional area. But yeah, it's true.
I think it's also like, I hope that it's encouraging, that it's like there's a lot of work to do, and this is just a tiny part of it, is the going to sea part. I know a lot of people that we love it, we all are doing this life, but I know a lot of people that have no interest in coming out on a ship, and like that is not for them for many reasons, whatever reasons those are fine, are of course totally valid. Whether it's a chapter of your life or your whole life, that this is kind of a disruptive way to live your life sometimes, you know, like, oh, I'm going to disappear
Triggered.
for 3 weeks and
There goes a ball.
head out
Thank
somewhere.
you. Copy that.
That, you know, understanding that there's a lot of ways to be involved.
Yep.
Jake, did it fire or?
Yeah, it fired.
Okay.
But I'm gonna have to print another ball.
Yep.
Yeah, there can be like,
Yep.
in the same way a lot of oceanography is done with satellite data, and you know, there's a lot of different data.
I can bring it home. Yep. Sorry, Megan.
No
Getting
worries.
called by Dan.
Sorry, did you say that both of them fired or just? Okay.
Science, what sample number is that?
Oh, someone's not speaking on SPL. Sorry, it's 46.
Thank you.
Yep. Yeah, if you could. Nav, if you don't mind, could you put a dash instead of an underscore so they're all consistent?
Well, I can. They're not going to be because we started with underscores, the cruise.
Okay. That's fine.
Favorite file convention. Round the room. Go.
You gotta update the Herc sample.
I mean, we should do that. Like, I mean,
Is that, we're doing that
Yeah.
now?
I don't know. Megan was doing it.
Yeah.
That's
Well,
what-
I'm now mad at Megan for switching from underscores to dashes. So
Well, in the sample data log, we always use dashes, so.
Yeah.
Yeah.
Oh yeah.
that's
All
one
right.
of those
Come on,
really
stop.
interesting
Yeah.
things that are also underappreciated is how important when you're dealing at the scale of one of these expeditions that little things like keeping that kind of thing consistent so it's machine readable and you can write scripts to do it
Wanna
and it
let Mike know
organizes the
that
same way with whatever
winch
file
cable
system
is-
you're using.
yeah.
You know, I've literally spent a week in a meeting with 5 other people arguing about how we were going to name samples for a long-term program. So it's an incredibly important aspect that is totally underappreciated.
And that's also like one of the hardest things to do. Like earlier today, we were messaging in our group chat, me and Pierre and Nicola, and we're like, how are we gonna name these samples?
And, you know, when you come back from an expedition like this with literally hundreds, sometimes thousands of samples, including subsamples, like going back and manually changing them is a really annoying thing.
Yes, and very tedious.
You can see a couple of these little clouds of ink or goo coming through. These are either disrupted, these are probably pseudomorphs, which are basically a type of ink produced by squids as a defensive mechanism. As the ROV comes up, it spooks them. There, you can see a couple squids and some of the other cameras. And as we're scaring them, they're producing these kind of gooey ink balls to try and distract predator.
Yeah.
I've seen
I have a feeling that all of our 100-meter replicates are just gonna be mainly
squid?
squid. Yeah.
Yeah.
I wonder if some of this sediment coming off of the ROV right now is going to end up in your eDNA bottles.
Almost certainly.
It's okay. And they'll probably question why there are like deep sea microbes in the middle of the water column, but note to make.
Well, it's good that we have the video to provide an explanation.
There's a siphonophore there as well.